Okay, our next presenter is Dr. Franck Dayan from Colorado State University. I've been fangirling over him all morning. He received his PhD in plant physiology at Auburn University in Auburn, Alabama in 1995. We should drop dates. I don't like telling people how old I am, so you should never do that either. Yeah, okay, anyway. He was a research scientist with USDA ARS, Natural Products Utilization Research Unit from 95 to 2015. He's now a professor of weed science and nutrient distinguished scholar of agricultural sciences in the agricultural biology at Colorado State University in Fort Collins, Colorado, where they do amazing work. Todd Gaines, Franck, the whole, it's an amazing group. Okay, sorry, not that Rob's not amazing here at the University of Manitoba, but wow. Okay, his research focuses on herbicide mode of action and mechanisms of resistance in plants. He serves on the editorial boards of Outlooks on Pest Management. He has published more than 200 research articles, 70 reviews or book chapters, and has more than 40 patents. This is the man. Tell us what you know.
Well, I hope I don't disappoint. Thank you for the introduction, and thank you for the invitation, Rob. I hope to get to talk to you a little bit more later. First, I want to thank you both, Rob and Martin, because they kind of set up the stage, and I had too many slides, and some of it has been covered already, so I'll be able to, if I skip a slide, it's because it's something that has been talked about before. All right, so I need to click on my screen, I guess, so it will do that. Here we go. All right, so I was asked to talk about a lot of things, and so I decided to cut it in 4 different sections to try to make it easier to handle. And so I will first talk about the status of ag industry. And some of that information is pretty well known already, but if you look at the cost of R&D through the years, it basically doubled since 1995, and now to get a product from discovery to registration is about $300 million, probably more. That's the most recent data that I have, and I'd love to hear from industry people what they know now, but it's probably more. And then you have a breakdown of the cost, where it goes from the chemistry, the biology, all the way through the ecotox, environmental, and then the registration process. Same thing, it used to, the cost to commercialization is shown here. And now companies spend about $3.2 billion globally to promote new herbicides. And if you look at the breakdown, look at the 2019 right here. So is the mouse working on the screen? No, it doesn't. Okay. It's amazing. Out of the $3.2 billion, more than half of it is in research, in discovery of new AIs, and in the development of new AIs. So companies really keep trying. We've not had much success, but companies keep putting a lot of money into the discovery projects. And this is a really interesting graph that shows by year on the x-axis and the number of new mode of action over by years. And so essentially, we used to have one new mode of action, so not new AI, but one new mode of action, a new way of killing a plant, one new mode of action every 2 years, and then it kind of stopped. And then for the last, 30 years almost, we've had very few new modes of action being introduced. And one of the questions is, why is that? Have we run out of target sites that are good for herbicides? And I don't believe so, but that's definitely a fact that we have to deal with. And I will show you later on what the industries and startup companies are doing to try to restart the discovery of new modes of action. This graph is also very telling. What we have on the bottom are years, and on the left axis is cumulative new herbicide AI introduced. So the number of AIs introduced, and that will be the square. And on the right side is the number of patents for new herbicides. And what you can see is that there was an increase of about 5.5 new AI per year for many years, up until a certain date, and now it's about 2 new AIs per year. And then the circle, basically the number of patents, there were about 100 patents a year for many years, and then all of a sudden they started dropping right here. And I just want to point that this graph was put together by Steve Duke, but that's the date when glyphosate-resistant crops were introduced. And there's a really good correlation that the number of AI patents have gone down a lot because at the time we were told that glyphosate will solve all of our problems. So we started doing research in that area. And so now we know that's a problem. We have resistance. We have to come up with new ways of controlling weeds. And so there's hope. There is hope for new modes of action. So I just wanted to kind of touch on how traditionally herbicides are discovered, because I want to then spend some time on some new approaches that are coming along. So traditionally, the ag-chem industry, and they still do it that way primarily, is a chemical-based discovery, where they synthesize a bunch of compounds, they test them in the greenhouse, they found leads, then they do lead optimization, which they go back to chemists and they do some small changes to the structure to make it behave a different way. And in the end, they'll take it to the field, they select those that are active, and you end up with maybe, if you're lucky, a compound that you can register. And as of the most recent data is 2019, companies have to screen 150,000 compounds to come up with one new active ingredient, right? And that takes $300 million and 11 years from discovery to registration. So it's a long, costly, very rare event that companies are looking for. They've tried to develop new methods to speed up the process, and one of the methods is high throughput target. What they've done is they've identified targets in the plants. They developed an enzyme assay that can do in the test tube, so then they can test lots of compounds very rapidly to see which ones are active on the target. So they have many hits, and then they refine the structures until they get really a good active ingredient in a test tube to inhibit a specific enzyme. And then so that's very short period of time. So that kind of takes away the 11 years of screening. And then they optimize the structure for optimized compound. That's one way that they try to overcome that problem, by not only going to the greenhouse, but going in the test tube, but not testing on plants, but testing on enzymes. Another way has been to look at the crystal structure of a protein, figuring out the structure of the binding pocket, like Martin showed, and then they design a molecule specifically to fit the pocket. And then there's an iterative process to go through these. Companies have spent millions, maybe billions of dollars on this. It's crazy. And this, by and large, has not been successful. They still went back to going back to the greenhouse to identify compounds. So what can we say? What's the result of all that work? So recent development. There are several new active ingredients that target known mode of action. So I'm not going to talk about those because there'll be the group 1 through the group 27 or whatever HPPD is. Now there are a few new modes of actions that have been discovered, but on old chemistry. So these were herbicides that were being used, but we did not know how they worked. So one of them would be endothol. See, this is the work that we've done when I was at USDA, where people had used endothol for, 50 years without knowing actually how it killed the plants. And we figured out that it inhibited an enzyme called serine threonine protein phosphatase. And it's an enzyme that's involved in regulating protein activity. It's part of a cycle with phosphatase and kinase. And this will not, maybe I guess it applies to Canada because it's only used really for weed management in aquatic systems. And I don't know if it's used in Canada at all, but it's a very effective compound. Another example will be the discovery of the mode of action of synmethylene, which now is called Luximo. So synmethylene will be the structure the most on the left. It's a very old compound that was, by the time I worked on it, it was DuPont, but maybe somebody else even discovered it. Well, it's not until 4 years ago, that there was a report from BASF that they figured out that it inhibit a new step in lipid biosynthesis. So that is a new target site, but it's an old chemistry. And then another example will be aclonifen. Aclonifen is a diphenyl ether that was actually listed as a PPO inhibitor, group 14, and it does inhibit a little bit, but the plants not only, the symptoms were not like a typical PPO, there was something different about it. But it turns out now we know that it inhibits a step in plastoquinone biosynthesis, so it causes bleaching, but not the way phytoin desaturase inhibitors do it, and not the way HPPD inhibitors do it. It's kind of in between the two pathways. So that's again a new mode of action based on an old chemistry. And then there are the few, the rare breed, right? New modes of action from new chemistry, and that's a product that this is cycloprimorate that was developed in Japan. And that also is a bleaching compound, as you can see on the picture. It's a compound that's being developed for rice, so it's probably not very applicable for Canada, but it is also an inhibitor of carotenoid biosynthesis by inhibiting plastoquinone biosynthesis, but differently from HPPD and differently from aclonifen. So this is really one example of, after all these years, of a new mode of action based on a new chemical structure. And then the other one will be the compound from FMC, totflupyramylate. It's really hard to say. I think they changed it to something maybe a little bit easier to say, Dodilex. And that is actually a new compound that inhibits nucleotide biosynthesis. And that also right now is being used on rice. It's being developed for rice, and I believe the release will be 2025 or 2026, so it's really coming up. And that will be maybe the most recent commercialized new herbicide with a new mode of action. So people ask always, and farmers are waiting for the silver bullet, how we're going to deal with all these resistance, how we're going to come up with new compounds. And so what we did last year, actually this year at WSSA in 2024, Dr. Duke, my former boss, and I, we organized a symposium where we invited 10 startup companies and people that use methods that are not typically used by industry to come up with new methods. So I just want to give you a quick report on that. So first there was several people that talked about natural products, and because I worked on natural products for 20 years, I was really interested in this. So I just wanted to kind of real briefly tell you why it's interesting to work with natural products. I talked to you about the high throughput screen the companies do, where they make lots of compounds, test on greenhouse, and try to find the compounds that have interesting activity and hopefully a new mode of action. And so they do very high throughput screening, very rapid, of many compounds over a short time. Evolution essentially is the opposite of that. What they do is they look at many chemical interactions, and we don't know, plants don't know what the chemical interactions are. They just are exposed to insects. I'm using insects, for example. It could be weeds, but there's beneficial interactions, the negative interactions. And through a novel pathway, through gene duplications, plants have evolved new pathways called secondary metabolism that can then enhance the positive response of positive interactions. So you have more pollinators coming to you, you have beneficial insects coming to you, or you produce compounds that will repel those that have negative response. And so in the end, you end up with a competitive advantage. And this then is the opposite of what industry does. It's many target sites over a very long time, but then it's giving you a chance to identify new target sites. So that's what I call the high throughput screening of life. And so based on that, a lot of people have looked at natural products for pest management. And that has been pretty successful for insects and for disease. But for weed management, it's not been very successful. And there's Steve Duke wrote this review a while back, last year actually, why there's not really good success of microbial bioherbicides. And he goes through, so I'm not going to go through all these, but essentially he breaks down the scientific, technological, and commercial hurdles that have to be overcome. And at every steps, there's issues with bioherbicides. That's the problem. However, it's not a lost cause. There's a company called ProFarm. It used to be a Marron Bioinnovation. They've been working on a compound that's being commercialized as opportune. And the problem they had was that for successful treatment, it was $2,000 per acre. And the reason is because the microbe that they had to make the product made that will be 50 milligrams per liter. So in a broth culture, in a fermentation chamber, the production was very low, so they were not able to generate enough of it. And so they've done a lot of work to optimize this, and they've did screening for new strains. And now they went from 0.05 grams to 5 grams per liter. So they increased by 100 times the production capacity. So now that becomes, that reduces the cost per acre and all of that. But that takes a lot of work. So there's a lot more we could say about natural products, but I wanted to mention that to lead into some other things. So, glyphosate is actually a natural product. So it's not sold as a natural product, but it is a natural product, and it's like $1 billion natural product. So I do want to mention it here because it was discovered from a microbe in the form of bialaphos, which is basically glyphosate attached to 2 amino acids. And in Japan, actually, bialaphos is commercialized as a natural herbicide. The plant takes in bialaphos, breaks it, removes the amino acid, and releases the active form of bialaphos, which is L-phosphinothrycine, which is actually glyphosate. So glyphosate is a natural product, but it's actually not commercialized as a natural product, and it's not approved for use in organic agriculture because the way it is made is it's made in a mixture of the L, the active form, and the D, which is the inactive form. And because the D is not natural, then that's no longer a natural product the way it's commercialized. Now there's new patents out there, there's companies that are actually commercializing only L. So that now instead of having to put, let's say, 1 kilogram of glyphosate per acre, or I'm getting my units, so grams per hectare, or that's the way I work. So let's say you put 1,000 grams per hectare of glyphosate. Now you can put 500 of the L form, and you get exactly the same activity. You put half as much because you only put the active ingredient in it. And the reason why I want to mention this is because glyphosate is a phosphonate. So it's a kind of an amino acid-like structure that has a phosphonate group right here. Okay, and why is that important? Because at the conference, that company called Macro MGX, their area is they focus on the discovery of phosphonates. And these are examples of phosphonates. On the top is the activity that they have. And the reason why they're active is because they mimic natural substrate. In the case of glyphosate, it mimics glutamate, the substrate of glutamine synthetase. So that will be the second from your right. So what they do now is they go directly in the microbes, look for the genomes, and look for the information about enzymes that are involved in the synthesis of this natural phosphonate. And they can screen lots of strains to find those that have the signatures for the synthesis of these interesting natural products. And they've gone through these and they've identified a gene cluster in Panteo ananatis. And what they found is that basically they retro-engineered this. They found the genes for the biosynthesis, they identified the structure, and now they have a way of growing this microbe to generate lots of these pentaphos, which is a natural phosphonate, which is very herbicidal. So if you want to know more about this, you can read the review that we published on that. So then it's looking basically at the genome to identify new compounds, right? And in this case, they were successful. Now, there's a paper in Nature that came out a few years ago where they used the same approach. And what they were looking for is they knew this asparagillus was making aspartic acid, and it's herbicidal. Well, they looked at the structure and they figured to get this molecule, you need these genes. And they went and they sequenced the genome, and they found genes that could encode for the pathway for that natural product. Now, the reason why that's a little bit different from the previous slide is that in addition to finding the genes for the synthesis of that product, they found a gene duplicated for a gene involved in amino acid biosynthesis. So now this microbe has the pathway for this toxin and a gene that's a duplicate gene for DHAD, which is part of the branch chain amino acid biosynthesis. And what they postulate is, why would there be two genes in there? And actually what they've found is that the compound is toxic to the organism itself. So the organism made a herbicide-resistant enzyme so it could survive. So using this method, that genomic approach, not only they have identified the biosynthesis of that compound, but they identified a gene that provides resistance. So you have already a pre-made gene that you could put in a crop to make the crop resistant to the toxin. So you get a new herbicide and a new resistant target to that herbicide. So these genomic approaches are very powerful, and especially in the area of natural products. I need to move on to another method. So this is a company I've been interacting with. It's a startup company in England, MOA Technology from Oxford University. And they do in between. So companies do greenhouse testing on a variety of weeds and crops. You have to have a huge amount of space to test these compounds. What this company does is they work with Markentia, which is it's a plant, but it's a macroscopic plant. So what they've been able to do is they can screen hundreds of thousands of compounds, but not on a target site, but on a small plant. And then they have a phenotypic method that's very advanced. And what they're looking at but determining what the plants look like, they can identify all the known target sites. And then if they have a phenotype that does not match a known target site, then they suppose that it's actually a new target site. And so they've discovered probably more than 60 potential new target sites, not in a test tube, but actually on small plants. And so they have a lot, so that's called the Galaxy, where they can screen all these compounds, and I think they've been around for, a few years, and they tested like 800,000 compounds already. Pretty amazing. And they've discovered, potentially more than 60 new target sites, and now they have a target platform that is to actually identify the target sites, and it's kind of complicated, so I don't want to spend too much time on this, but the review talks about how they do that as well. Another approach, which is really interesting, is PROTAC. So traditionally, the way we look at a herbicide is we have a target site and we put a chemical compound that binds to the target site and keeps it from functioning. What Earth Bio does is PROTAC, where they identify a target site, and what they do is they use the plant machinery to have the ligase, the machine that breaks down protein in plant, to target the target site. So what happens is they identify the target site. What they do on the bottom, they identify a bait and a linker that attach the linker to the ligase, and the ligase then is connected to the target protein. Then that recruits ubiquitin, and the protein then gets ubiquinated, and that's a signal in plants to say, okay, this protein needs to be destroyed. So then it goes through the proteasome, and then the protein is completely destroyed, and then that gets recycled. So you can get your PROTAC binder that basically it's the linker and the bait. The linker and the bait go in the plant and then put the ligase and the protein is in contact, and then that targets that protein for degradation. So they show that it works. Now, we're not anywhere near having that working as a commercial herbicide, but it would be really a new way to kill plants. And so that's one exciting new area for potentially new way of new herbicides. Another way is that's a company from Israel, Progeny. What they've looked for is proteins that are what's called undruggable. So on the left side, you have a protein like a typical herbicide target, where there's a binding domain. You can put a herbicide in there that keeps the enzyme from working, and that kills the plant. Some proteins, you don't have a good place to put a herbicide. So what they've done is looking at proteins that require interactions with other proteins. So for a pathway to function, sometimes two proteins have to come together. And what they decided to do is, can we disrupt that interaction? And that instead of inhibiting the enzyme, we inhibit the pathway by preventing the proteins from interacting with each other. And they've actually been successful. They found these two compounds here. So the green will be the control, and the white will be the treated plants in the petri dishes here. So these compounds are herbicidal. And what they found is that these compounds prevent the synthesis of cysteine, which is a key amino acid in plant, by preventing the interaction between the dark blue protein and the light blue protein. I don't want to talk too much about it, but essentially those compounds bind where those proteins interact, and those proteins no longer can interact, and then you stop the pathway. So that's maybe another way of targeting new mode of action that were not able to be targeted with traditional herbicides. Another company, Encochem, in Massachusetts, they're using what's called DNA-encoded libraries, and this is kind of amazing. What they do is they synthesize billions of compounds in combinatorial methods, and they attach pieces of DNA so that every molecule has a code, like a zip code for it. And so now we're not talking about hundreds of thousands of compounds, we're talking about billions of compounds. Now they have a target in a test tube, and then they can test all these billions of compounds. And so they have a stepwise process. They show that it works because they were able to identify a new PPO herbicide, for example. So it's not a new target site, but they were able to use this method to identify PPO as a target site. So now they are looking for using this method to, and so because it was a known method, they were able to talk about it at the conference. They have a lot of new target sites that they're not able to share with us that are under development. Then the last company I want to talk about is another one from Israel called Agronomists, and I've worked with them. And so this is completely, the front end is completely computational, where they can screen billions of compounds, and they have an algorithm called AI4AI, which I thought was a pretty clever name, Artificial Intelligence for Active Ingredients. I told them to patent that name, register that name, so it was a pretty clever name. But anyway, so what they do is based more on the structure of the chemical themselves, and then the crystal structures of all the known proteins. Especially with some of the new methods, now we have access to crystal structures or models of crystal structures of many proteins, and they're able to come up with new target sites through this method. I've worked with them. They've identified a PPO herbicide. So we published that, and they've identified a new PS1. It's a natural product that inhibits photosystem 1, but it's not toxic like paraquat. So we'll see what's going to happen with that. But the bottom line is there's no silver bullet, because all of these technologies are probably 10 to 20 years from in the future, right? Before anything will actually come out of that. So people are working on it, but it doesn't solve a problem. So then I was asked to talk about herbicide resistance a little bit, but since Martin did a lot, I'm going to skip a lot of that, right? But then I'm going to give you some examples. Specifically, I was asked to talk about Palmer or water hemp and kochia, because we were at Colorado State, we work a lot on these plants, as you can imagine. So I'm going to go through these slides. First, that herbicides don't cause mutations. They select for resistance, and it's an evolutionary problem, evolutionary process. I'm just going to go about that. Just the only thing about this slide is, the term survival of the fittest was not invented by Darwin. Actually, somebody that read Darwin's book that's came up with the term survival of the fittest. And so much, Darwin liked it so much that when he did his second edition of his book, he started using that term. But essentially what it means is the plant is better, evolution, generally speaking, but in our case, herbicide resistance is making a plant better designed for its immediate local environment. So we're not talking about macro evolution, but micro, very local, right? And plants are very creative. So some of you may have seen this, but if you've not, I'm glad I can share that with you, because plants will evolve resistance to anything. So here is something that was reported from Asia, where on the left side you have a rice plant, on the right side you have barnyard grass. And the main difference is the barnyard grass, the leaves are more curved, all right? So through centuries, probably millenniums, farmers will go in the field, And they'll say, okay, that's a rice seedling, that's a barnyard seedling, so I'm going to pull the barnyard seedlings. And over thousands of years, now they've selected a barnyard plant that looks like a rice plant. So essentially, rice has evolved evolution to hand weeding just by changing the way it looks. That's the selection pressure of hand weeding changed the way the plant looked. Another example is some of the new harvest weed seed control. You're probably familiar with these. It's a really great method of intercepting the weed seeds in the chaff and destroying them so they don't get back in the field. And a study has been done where they've done it over several years, over and over and over. And so what you have here is on the x-axis is the days that it takes for the plants to flower, and on the y-axis is the percent of plants that are flowered. And what they've shown is the black line is the original population. And as they kept using the harvest weed seed destroyer machine, they selected for plants that flowered before harvest. And so the seeds were shed before harvest. So they basically selected the plants that already had seeds in the ground by the time of harvest. So essentially, evolution, these plants have evolved resistance to seed destruction, right? So we're fighting a battle that we're never going to win. We just have to be creative in addressing it, but we're never going to win this. And then this one is really well known from France, from Christophe Delis, where he was looking at sequences of black grass, and he's found a black grass sample from 1888 in a herbarium that already had mutations for FOPs and DIMS resistance. And of course, there was no herbicides around, so as Martin said, these mutations were already present. The herbicide just selected for the resistance. So then these plants have evolved resistance, essentially before resistance and before herbicides even existed. So then I'm going to move pretty quickly. It's across all continents and across cropping systems. So there's no systems or country that are immune to herbicide resistance. So now, let me talk about types of resistance. So briefly, Martin talked about that. There's non-target site resistance, and there's target site resistance. How much time do I have?
Oh, like 15 minutes.
Good, then I'll slow down. Actually, is there a question on what I've talked about already before I move on to this, since we have time? Any questions online or in the room? Yes?
Of all those new methods, is there any one that you think is best?
Okay, wait, can you
I'll
repeat?
Yeah. So the question is, of all the new approaches that I discussed, are there one better than the other?
Yeah.
So I think so. In my view, something that's fully computational will generate a lot of data, but you're working in a very abstract, will it have a target site, and will that target site actually be a good herbicide? That's probably the least, likely to be successful. The closer you are to the plant, the better it is. So, for me, the MOA approach, where they actually screen lots of compounds very rapidly, but not on an enzyme, but on actual plants, is maybe the best in between, where you can have the high throughput screen, but you're still testing it on plants. And Markentia is only a very simple plant, small genome. All the genome is completely sequenced, and it's easy to do mutations. So what they do, since we have some time, is once they have compounds that are active,
We don't have that much time.
and give a new phenotype, then they can do mutations to find mutations that cause resistance, and then through the mutation they do reverse genetics and can identify the target site. So I think that's probably the most powerful of them so far. Okay, so as far as I'll take a different take on ways plants can become resistant. So there's non-target sites, and then there's target site resistance. Sorry. So the way instead of going through my slides, when I teach my students, I make a triangle. If you think of a triangle, so if you're online, I'm sorry, maybe this camera, if you see me, there's a triangle. What you can imagine, on one top, you're gonna have the target site. On the bottom, now I wanna make sure I get it right. I should have been prepared for this. So, as I talk about it, it's going to come back to me. But essentially, the balance is how much of the herbicide was in the plant, how long it stays in the plant, and how quickly you inhibit the target. So yeah, so you have metabolism, you have absorption, metabolism, and target. Okay, so that's my triangle. Target is what needs to be inhibited. Absorption is the herbicide is applied outside the plant. It has to get into the plant. And metabolism is how long it's going to stay in the plant. Most herbicides, other than those for transgenic crops, are selective herbicides, meaning that they kill plants because the crop survives the treatment and the weeds die. And the reason why the crop survives the treatment is typically because the herbicide either does not inhibit the target. So in the case of group 1, broadleaves, the target is not inhibited. Or in most cases is the crops will metabolize the herbicide faster than the weeds can. And so if you have that triangle, and you think of those two mechanisms of potential resistance, non-target sites is you make the herbicide, the weeds change in the way they can metabolize the herbicide, so the herbicide is no longer present long enough to inhibit the target. Okay. And in the case of target site resistance, it doesn't matter how long the herbicide sticks around. If the target has a mutation, then it's not going to be inhibited. So you basically destabilize that triangle, right? So in a way, that's the best way to view this. And so as far as non-target sites, there's different ways that can be done. So I'm not going to talk too much about this. But mutations, enhanced metabolism is typically the most common mechanism of resistance for non-target sites. And for target sites, I'll just go through these illustrations. Typically, you spray a herbicide. Let's say there's 3 molecules of the herbicide and there's 1 target, where you inhibit the target and you inhibit your plant. Now, if you have a mutation, then that herbicide will no longer bind there, and so the target is resistant. So that's a target site mutation. Or you have multiple copies, as what Martin was talking about, where you put the herbicide, you still have 3 molecules of the herbicide, but now you have 8 targets. So you kind of bind all the herbicide, and now you have a bunch of targets that are not inhibited, and your plant survive. That's basically very quickly how that works. So what do we know? Some kochia, that plant is such a terrible for us in Colorado. So UPL has developed a product called Scorch that has 2,4-D, dichloroprop, and dicamba mixed together. Okay, and because we know we have some dicamba resistance, we did a screen for this, and what we have for dicamba, in the sense it's susceptible, is the green line. Dicamba basically accumulates in the plant over time. The red line is in the resistant one. What you can see is that dicamba does not accumulate in that plant. So the way that plant is, that kochia is resistant, is metabolism based. It just metabolizes the herbicide. Well, it turns out that it's already resistant to dichloroprop, and it's also resistant to dicamba. Oh wait, 2,4-D, sorry. So dicamba was on the left. It's already resistant to 2,4-D, and it's also resistant to dichloroprop. So essentially, the genes that are turned on to metabolize dicamba can also metabolize 2,4-D and can metabolize dichloroprop. And that's because, 2,4-D and dichloroprop are very structurally related. Kochia can evolve resistance through metabolism. These plants were not resistant to fluroxypyr, which is also a group 4. So now we have another population that is resistant to fluroxypyr. And what a student, Jake Montgomery, found is that there was a mutation in the day ground, and I don't have too much time about this, but that's in the group 4 herbicides. What you have is you have an auxin protein that binds to this receptor. And the way they interact is because auxin, the hormone auxin, binds there and makes them bind together. And these herbicides mimic the auxin, and they cause those two things to bind together, and that causes deregulation of pathways, and that's what kills the plant. Well, what we found is that there's a mutation in the degrowth, which is the key part of the protein that's involved in that interaction. And then that specific mutation that I'm pointing with the arrow changes the structure of the protein, kind of the structure that what Martin was showing, where now the two proteins don't interact anymore and the plant becomes resistant. So in the case of kochia, in group 4, it can evolve resistance through metabolism, or it can evolve resistance through target site mutation. Okay, so plants are just so flexible. Now let's move on to this is kochia in perfect. I'm almost done. Yeah, this is kochia resistance to glyphosate, and what we show in the middle graph in the pink is the number of the copy of the enzyme that's inhibited by glyphosate. So it ranges from 5 to 20. Some plants normally they have one gene copy, and these plants that are resistant, they can have up to between 5 and 20 copies of that. And then in blue line is the offspring of these plants. So we've done a kind of population study, and essentially it's the same range, and it's the same kind of mean number, so about 12 copies. On average, kochia plants, instead of having one EPSPS, have 12, and we know that if they have more than 3, they are resistant. So that shows that the parents are resistant because they have multiple copies of PSPS, and it's inherited. The offsprings are exactly the same. They have the same kind of, on average, the same number of copy number. And on the right graph, what we've done is we've done marker studies. We're looking at the type of EPSPS in the parent versus the offspring using markers. And what we can show is that in kochia, almost at least 80% of it is through cross-pollination. And then the red bar is the number of plants where we had no proof that it was cross-pollination, so we call them self. But it could be that it was a cross-pollination of plants that have the same genetic structure, so we could not detect it. So most of the inheritance of resistance is through cross-pollination in kochia. All right. Water hemp can have resistance to 2,4-D through metabolism. And this has been published, so the paper is on there. Is that presentation online? So the PDF, so all that's there. We have this fancy drawing that kind of summarizes all this, so I'm not going to go through this. But the reason why I want to talk about this is so we know that 2,4-D resistance in water hemp is metabolism-based. I've been working on a palmer amaranth resistant to dicamba, a population from Tennessee, from Tennessee, and so you have lots of resistance, 12x resistance and survival. You basically can't kill the plants, even at 1,000 grams. And we've done metabolism, and this is what the graph shows, that basically the plant metabolizes it the same way. So we know it's not metabolism-based. We've try to reverse the effect with malathion and with an inhibitor of GST or P450 monooxygenase, and we were not able to reverse resistance. So we know the resistance is not non-target site. Then we looked at transcriptomics, sequenced everything. There's no mutation that would provide resistance. So what we did more work, and what we found, so this graph on the left is a summary of, in green, genes that were overexpressed when the plants were treated with dicamba in the sensitive, and in the red, they were overexpressed in the resistant one, okay? And so those that are both overexpressed in the sensitive and the resistant, it's probably not very significant, but what you can see is some of them are only overexpressed in the sensitive, but the resistant one did not respond. And I just want to point out to this top one right here. That's the IA29. Because what I found a paper that says that IA29 is regulated by an enzyme called PIF4, okay? And then I told my student, well, look at what's the gene expression of P4. I had never heard of P4. And what you can see, P4 is overexpressed in green, in the sensitive, and it's not overexpressed in the resistant one, okay? Now, why is that important? Is because we found a paper that shows that if you have a mutation in P4, then you don't have epinasty anymore in plants. And the IA29 no longer responds. So in these plants, what we think is P4 is no longer responding, So I-29 is not responding, and the plant doesn't have any epinasty. So I just want to put a plug, if you're interested in some of the stuff we do. I'm teaching a plant biochemistry and agriculture online this spring, and then we have, oh, that's wrong. Anyway, so if you want to take classes at CSU online and hear from the best, not me, but Todd Gaines.
Everyone is the best that's been here so far this morning. Thank you so much, Franck.
Thank you.
While I'm getting you your gift, if we get all of these technologies up and going, will we be able to start manufacturing natural or synthetic or whatever herbicides as quickly as we used to if we get all of your magic companies working full force?
I think the synthesis side of things, I think that's not an issue anymore. I think we have the methodology. It's just really identifying the new target sites and the new chemistry is what's the limiting step right now.
So in 10 to 20 years, we can get back into full swing?
But so this is go back to my first line, the tail wagging the dog. The weeds are a problem. The herbicides were a solution, and herbicide resistance is, wagging a dog, right? And so I think I like, the conclusions that have been said so far is that really integrated weed management is where we need to go, because doing the same thing we've been doing, we're going to have the same problems over and over and over, and we know that.
Thanks, Franck. I appreciate it.